Hello friends, in this lecture we will talk about germplasm and how its conservation is done. I am preparing this PPT for your sake so that we can understand this germplasm, how important it is to be conserved. All these things we'll study in this lecture. So basically, what is germplasm? How it is conserved, what are the different types of conservations of germplasm, all these things we'll study in this lecture. So let's begin with germplasm and its conservation. So if we see the definition of germplasm, germplasm is a live source of all the genes such as seeds or any plant parts which can be conserved for very long period of time, and it can be regenerated whenever required in future. Means basically germplasm is a future plant or future life that we have to conserve today. Okay, so if you take a simple example, seeds can be a germplasm, because seeds, if you preserve the seeds, that seeds will can grow years after years. Okay, so this can be a germplasm. Even the animal cells, plant cells, because we know tissue culture technology is there, and because of tissue culture technology we are able to generate an entire plant from a single cell, though, so that single cell can be a germplasm in case of plants. It is not possible in case of animals. Okay, so what are the different characteristics this germplasm is having? It contains all the information of that organism. Okay, total information of that organism is present in that germplasm, and it is very imperative or indispensable for us to conserve the germplasm for the sake of conserving the biodiversity, genetic diversity, because if we, until and unless we conserve the germplasm, and if we get that germplasm get extinct, so in no any manner we are able to recover that germplasm. So germplasm conservation is very important thing as far as present scenario is concerned. And then it provides a gene pool for different genes which act as a reservoir. So for the breeding purposes, plant breeding purposes, hybridization experiments, this conserved germplasm can be a gene pool. Means we can take a particular germplasm from of one plant and germplasm of another plant, when we can do the hybridization or we can do the breeding. Okay, so it's very important to conserve the germplasm. It is very much important in case of rare species, endangered species, because these species, if we don't take the care of plants, these plants will get extinct in very near futures. For example, critically endangered species. So to preserve that, to conserve their genetic diversity or their diversity, it is very important to conserve the germplasm. So germplasm can be conserved of cultivated species, wild species, land races, or breeding lines. So basically this germplasm conservation is of two different types: in situ conservation and ex situ conservation. So what is the difference between this in situ and ex situ? In situ conservation means conservation in natural habitat. In natural habitat means wherever it is naturally growing, if we are able to conserve that particular plant over that place only, it is considered as in situ conservation. And if we bring that plant from its original place and if we cultivate to some other protected area at our place, then it is not at their natural habitat but at some other place. So in that scenario, that kind of germplasm becomes ex situ conservation. For example, if we take, we'll start with in situ conservation. In situ conservation, as I told you, that on-site conservation of germplasm is called as in situ conservation, or the preservation of germplasm in their natural habitat is called as in situ conservation. We can take example of in situ conservation as biosphere reserve, national park, wildlife sanctuaries. All these protected areas comes under the in situ conservation because we conserve them at their natural habitat in wildlife, in biosphere reserve, national plants means we go there and we conserve there in their natural habitat. So it becomes in situ conservation. So you can see Jim Corbett National Park, Nilgiri Biosphere Reserve, Bharatpur Bird Sanctuary. All these are what? These are the in situ conservation method. For example, second is ex situ conservation method. It is considered as a off-site conservation of germplasm that is outside their natural habitat. It is termed as ex situ conservation. Example of it is gene bank is there, seed bank is there, botanical gardens are there, then zoological parks are there. Here, the germplasm is conserved outside their natural habitat. Okay, so gene banks, it can be in vivo, in vitro. All this stuff comes under the ex situ conservation. So you can see here seed banks are there where seeds are preserved at one place, or gene bank, tissue culture. All these things comes under the ex situ conservation. I hope you understood the what is in germplasm, what is the importance of germplasm, and what are the different types of germplasm. So thank you very much.